Let's go into the second part of regulation of transcription. We will start talking about how it works in eukaryotic systems, and of course we will focus on the human system. So as I said before, it's more complex in eukaryotic systems versus prokaryotic systems. And last thing I said is how transcription factors and regulatory proteins interact with DNA. So you have amino acids in proteins, and you have the nitrogenous bases in the DNA in a certain sequence for both of them, certain sequence of amino acids, certain sequence of bases in the DNA, and that's how they specifically interact with each other via non-covalent interactions. So there are different mechanisms and diverse mechanisms in eukaryotic systems. Okay. So it's more complex, but the same basic principles apply, except that, well, there are exceptions. Okay, so transcription in eukaryotic cells is controlled by cis-acting elements, and there are more types of cis-acting elements in eukaryotic genomes versus prokaryotic genomes. So in eukaryotic cells, in their DNA, there are promoters. There are proximal promoter elements. There are multiple promoters, as we mentioned before. There are proximal promoter elements that sort of like synchronize transcription of genes that participate in similar mechanisms. Okay, it's like, as we said before, it's like an alternative of operons in prokaryotes. There are enhancers and there are also silencers. Now, in terms of transcriptional regulatory proteins, it's basically, as you will see, there are more regulators that can function as activators and repressors. Each one of them has a certain function. They participate in chromatin remodeling. They participate in DNA modification as well. And we have non-coding RNA molecules. We don't know what many of these RNA molecules do, but it is a fascinating area of research now that we have discovered that there are so many non-coding RNA molecules. They have a lot of functions that we don't know about, and there are many types of non-coding RNA molecules. Again, when we say non-coding RNA molecules, we're talking about RNA molecules that are produced by transcription from certain regions in the human genome, but they are not translated into protons. So the idea here is that you have a gene, okay, and this gene has a transcription start site right here, and you have multiple regions upstream of this gene. They can also exist downstream, like enhancers, for example, and certain types of promoters, and they bind to, and regulatory proteins would bind to these regions specifically, non-covalently, and they can regulate transcription. So right here, for example, you have an activator that would induce little transcription, these activators would bind to DNA, inducing a lot of transcription. If one of them binds, there would be little or no transcription, so having more of them would induce a lot of transcription. If you have a repressor that binds to a certain DNA region, it might block transcription altogether. So that's the whole idea behind it. Okay, now, so how do transcription factors regulate gene expression? Okay, so what transcription factors do is that they cause what is known as epigenetic or epigenomic changes in DNA. So what do we mean by epigenetics or epigenomics? So epi means above or in addition to. It's something that is at a higher level of regulation than the regulation that we've talked about in prokaryotic cells. So it's a higher level, and that's what epi means. So basically, when we say epigenetics, it's basically genetic alterations that regulate gene expression without changing the DNA sequence. Okay, so you have what is known as this can happen via chromatin packaging. We talked about heterochromatins and euchromatins. We'll talk about it again. There are chemical modification of histones. We'll talk about that. Or the DNA itself, or the bases in DNA, can also be modified chemically as well. So we'll talk about all of these mechanisms. So if we look at the structure of transcription factors, they have at least two domains. What do we mean by a domain? So a domain is basically, you have a protein, and a protein would have a three-dimensional structure. Now, part of the protein would have its own three-dimensional structure. Okay. Now, this domain or this structure would form independently of the rest of the protein, and it usually has a function. Now, what do I mean by it forms independently? It means that if we remove this domain, if we cut it out of the protein, it can still maintain its structure, its three-dimensional structure, and it can also maintain its function. Okay, amazingly, huh? So, so it can be separated from a protein and still be functional. Okay, so that's what a domain is. Now, these transcription factors would have at least two domains, and we're talking about those that induce expression. So the positive, the activators, the positive transcription factors. So they have at least two domains: a DNA binding domain and an activation domain. A DNA binding domain is, well, as we've seen with CAP, for example, in the lac operon, or the in terms of the CAP. That's a good example. Well, it can bind to a certain region in DNA. So the idea is that, again, if we cut this domain out of the transcription factor, this structure, this three-dimensional structure, can still bind to DNA. Okay? So there are two domains: a DNA binding domain and an activation domain. So what is an activation domain? So basically, again, this is a structure of a transcription factor. It has two domains: a DNA binding domain, so it binds to DNA, and it has another domain that is an activation domain. Now, what this activation domain does is one of two things, or maybe both at the same time. What it does is that it can interact with other proteins, like the mediator that binds to enhancers or other transcriptional factors, and or it can modify the DNA structure, more specifically the structure of chromatin. Okay, look at the structure right here. This is a good drawing of a transcription factor. The idea is that you have two domains. So if you cut the protein from right here, this can still function. It can still interact with transcription factors. Okay, it can still modify chromatin, but what makes it functional at a certain location regulating a specific gene is this DNA binding domain. So this DNA binding domain brings the transcription factor to a specific sequence in DNA, and then the activation domain would do whatever it does. Okay, so that's how this would regulate a specific protein, a specific gene in terms of expression. Okay, so if you have the activation domain by itself swimming all around, well, it can interact with the same proteins that it normally interacts with, but it doesn't have any function because it's far away from where the gene is. Okay, now, how about eukaryotic repressors? How do they function? Well, there are two types or two mechanisms by which they function. The first thing is that you can have a repressor that has a DNA binding domain, but it doesn't have an activation domain, or it doesn't have a protein binding domain. Okay, so it only has this. So what it does is that it occupies this region right here, preventing the activator from binding to DNA. So there's competition between the two. Now, the other mechanism by which repressors function is that they bind to DNA and they do interact with proteins, except that they hold them in their place. The repressor prevents the complex, whatever the complex is, pre-initiation complex or certain types of proteins. So it glues them to their place. It prevents them from moving forward. Okay, so these are the two mechanisms by which repressors function. Now, so how do transcription factors regulate gene expression? As I said, one mechanism is by modulating the structure of chromosome. Now remember that DNA exists as a chromatin, and this chromatin, there are two types. You can either have euchromatin or you can have what is known as a heterochromatin. The difference between the two is that in euchromatin, DNA is relaxed, it's loose. Heterochromatin is basically when DNA is highly packed. So right here, this here, you have a heterochromatin. DNA is highly packed. So when a transcription factor comes in trying to bind to DNA, it cannot, because the sequence that it binds to is hidden. It cannot find it. So it cannot regulate, it cannot induce gene expression. So heterochromatin contains basically inactive genes. Euchromatin, on the other hand, is, well, it looks like this, where the DNA is relaxed. It is exposed. So the sequences that transcription factors bind to are exposed. These regulatory factors can bind to the DNA easily and they can induce gene expression. Okay. Now, what regulatory proteins then do is that they switch DNA between the two structures of chromatin. Euchromatin for active genes and heterochromatin for inactive genes. Okay, so that's what we mean by modulating, modifying, changing the structure of chromosome. Now, how does this happen? How do these regulatory proteins change the structure of chromatin? Well, one type is known as chromatin remodeling factors. Okay, so remember, when I say factors, I mean proteins. So what these proteins do is that they remodel, they redecorate, they change the structure of chromatin. And they do so by one of three mechanisms. They either remove histones altogether from DNA. Okay, so you might have histones wrapping DNA around them, as in nucleosomes, but they are removed. So you have a lot of them in here, but then these histones are removed, exposing DNA. So that's one mechanism. Another mechanism is that they reposition nucleosomes, making DNA accessible. Like here, for example, what this does is that it pushes this histone, this octamer right here, it pushes it away, exposing this DNA right here, exposing the promoter region, allowing for the RNA polymerase to bind to the promoter. Or what they can do is that they alter, that is, they change nucleosome structure, allowing protein binding to DNA. Okay, so I'll give you an example right now in the next slide. So what chromatin remodeling factors do then is that, themselves, by the way, chromatin remodeling factors, they can be associated with transcriptional activators and repressors. So themselves, the transcriptional activators can by themselves be chromatin remodeling factors, or they can bind to DNA and they can recruit, that is, they can attract chromatin remodeling factors to that region. So here's an example of how the structure of chromatin can be modulated. Notice here, right here, we have a heterochromatin. So DNA is highly packed. Remember, we talked about histone H1, right? And we said that histone H1 forms a chromatosome. And it's found, the histone H1 is found when DNA is highly packed. Okay? It's not part of nucleosomes. It gets removed from a relaxed DNA from nucleosomes. So what happens here is that you have a protein known as P10. P10 is actually a tumor suppressor gene, meaning that it prevents the formation of tumors. Okay, and it does so by inactivating genes that induce cells to grow and proliferate and divide. Okay, so that's what P10 does. So P10 interacts with histone H1 and it brings it to DNA and it allows for the formation of heterochromatin. So certain genes would be inactivated. Okay, genes that are regulated by this protein P10. If P10 is removed, then H1 would be released and DNA is not packed anymore. It looks like a euchromatin and genes would then be induced. So what P10 does again is that it binds to DNA at certain sequences, regulating or inhibiting gene expression. What genes? The genes that induce cells to grow, divide, and proliferate. Okay, so what P10 does then is that, or the way it's regulated is that when it is released, okay, when it gets inactivated, H1 would then be removed right here. H1 is removed and DNA becomes more relaxed. And this gene would be induced, it would be activated, it would be expressed, okay, allowing cells to grow and proliferate or grow and divide. Okay, so this is a mechanism by how transcription factors can modulate, change, modify the structure of chromatin. Okay, so what else? What happens is that you can change the compactness, the packaging of chromatin. And this happens by chemical modification of histones. So one mechanism is, again, by chromatin remodeling factors, changing the compactness of the DNA by changing the position of histones or the association of histones with DNA. The other mechanism is that by chemically modifying histones. Okay? And this happens via a number of chemical modifications. You can have acetylation, you can have methylation, and you can have phosphorylation. And this can also be mediated by non-coding RNAs. Okay, so what do we mean by chemical modification of histones? Well, one type of modification is acetylation. That is the addition of an acetyl group, which is right here. This is an acetyl group. And this acetyl group is added to an amino acid that is found in abundance. That is, it's found in large quantities, large number in histones. This amino acid is known as lysine. Now, lysine is a positively charged amino acid. Okay? Remember we said that histones are positively charged because, and that's how they interact with DNA non-specifically, that is regardless of what the sequence of bases is. Well, this is one of the amino acids that histones are rich in. It's lysine, it's positively charged. So when lysine exists in the ionized form, when the positive charge is exposed, it has very strong interaction with DNA. So making the DNA compact, making it in the heterochromatin structure. Now what happens is the acetyl group is added to the positive charge right here by an enzyme known as histone acetyltransferase. So it transfers an acetyl group to histones. Histone acetyltransferase. So the acetyl group is added to lysine. Okay. So what happens here is that now the positive charge that exists in lysine is removed. It is masked. It's covered. Okay. It doesn't exist anymore. And that loosens, it weakens the interaction between histones and DNA. So DNA becomes more relaxed, more in the euchromatin structure, and that induces gene expression. Okay, so histone acetylation by enzymes known as histone acetyltransferases. Now the opposite is true. That is, repressors can also have an enzymatic activity, okay, known as histone deacetylase. That is, histones that remove the acetyl groups. So what they do is that they remove the acetyl group, exposing the positive charge, strengthening the interaction between histones and DNA, making DNA in a heterochromatin structure, so that would inactivate gene expression. So histone acetylation is an example. Okay, so here you have a heterochromatin again, and what happens is that you have acetylation of histones, and that would weaken the interaction, making DNA relaxed, and that would activate gene expression. Okay, so interestingly, transcriptional activators and repressors are associated with these enzymatic activities. So activators would have acetyltransferase activities. So what they do is that they acetylate histones, weakening the interaction between histones and DNA, and repressors have deacetylase activities, meaning that they remove the acetyl group, converting DNA into a heterochromatin structure. And interestingly, transcription factor 2D is associated with a histone acetyltransferase enzyme. And this is why transcription factor 2D is the first one that comes to a promoter of a particular genes, relaxing DNA and activating gene expression. Okay, now there are other modifications of histones, and these modifications include methylation and phosphorylation. Now, the effect is not as clear as acetylation. In general, phosphorylation would induce gene expression, but in terms of methylation, the effect is dependent on where this modification takes place. Sometimes methylation of certain regions of DNA would induce gene expression depending on where this modification takes place. In other cases, modification by methylation would actually repress gene expression. So it depends on where this takes place. So right here you have histone molecules, and they have tails. Okay, if you've noticed this representation right here, histones have tails, and these are the tails that get modified by phosphorylation, methylation, or acetylation. Okay, so that's all I want you to know. That is, histones can be chemically modified mainly by acetylation, and we talked about the effect of acetylation, but they can also be modified by methylation and phosphorylation, and the effect is dependent on the type of modification and where this modification takes place. Take home message, that's it. Okay, now how about non-coding RNA molecules? Well, I'll give you an example because we don't know much about these non-coding RNA molecules. But what they do is that you have this RNA, and what it does is that it can anneal, it can hybridize to a certain sequence of DNA that is complementary to the non-coding RNA. Now, remember how we said that RNA molecules can have they can fold, they can have a structure of themselves that is unique for themselves. Well, this three-dimensional structure of RNA molecules may actually fit with proteins. Okay, so it would attract these proteins to this specific region of the DNA, again, either activating gene expression or inactivating gene expression, depending on what the protein does. So this is one idea of what these non-coding RNA molecules do. Okay, so here's an example of how important non-coding RNA molecules is. Now, we'll talk about X chromosome inactivation as an example. So what is X chromosome inactivation? Now, we know that females have two X chromosomes, right? XX. Males have only one X chromosome. So in this case, females would have double the number of genes versus males. So does that mean that they express double the amount of proteins from these genes when compared with males? No, because what happens is that during development, one of the X chromosomes can be inactivated. So that's in females. So females would have only one active X chromosome, and males would also have one active X chromosome. So which X chromosome is inactivated? Well, it depends. It's a random process. It can be the maternal X chromosome or the paternal X chromosome. So it's like a mosaic, فسيفساء. يعني if you look at cells in a female body, you would find that some cells have paternal X chromosome active. In other cells, you would have the paternal X chromosome active. Okay, so even within the same organ, you would have this group of cells having this X chromosome active, and you have the other group of cells having the other X chromosome active. Okay, so how is X chromosome? How does X chromosome get inactivated? By long non-coding RNA. Okay, so it's basically one type of non-coding RNA molecules. It's called long non-coding RNA, link RNA, and this is transcribed from the X chromosome itself, the X chromosome that is to be inactivated. So you have transcription of the long non-coding RNA molecules from this gene known as ZIST. Okay, so you have production of a lot of RNA from this ZIST gene, and what this RNA does is that it coats this X chromosome. When it coats it, it inactivates it. It prevents transcription from all of the genes. And what happens then is that histones would be hypoacetylated. That is, in other words, they become deacetylated. They become, all lysines would become charged, and the interaction between histones and DNA would be very strong. Okay, and that would shrink. It makes the chromosome smaller. So this shrinks the X chromosome, and it becomes really a shrunk chromosome inside the cell. Okay, so this is the mechanism of X chromosome inactivation, and it's by epigenetics. It's by histone modification as well as DNA modification, as we'll talk about in a second. Okay, well, so DNA can be modified chemically like histones. Well, yes, DNA can be methylated. Remember, we talked about methylation of cytosine. So cytosine can be methylated, right? And I said that if cytosine is methylated, it becomes uracil. If methylcytosine is, so if cytosine is methylated, it becomes, I'm sorry. If cytosine is deaminated, it becomes uracil. If methylcytosine is deaminated, it becomes thymine. Okay, so what happens in DNA is that cytosine can be methylated. Okay, so that's a normal process. And this methylation takes place in what is known as CpG islands. What we mean by that is that there are sequences that are rich with C's and G's. And you have this region right here containing a lot of a lot of C's and G's. Okay, and that's why they're called islands. Now, usually these sequences exist upstream of core promoters. So when DNA is methylated, that is, when the cytosine is methylated, genes become inactive. Okay, so what happens is that DNA methylation induces the formation of heterochromatin, inhibiting gene expression. Okay, so that's DNA methylation. Now, DNA methylation is very important for a process known as genetic imprinting. So genetic imprinting is a term that indicates the following. Some genes can be imprinted, meaning that they specifically, any one of us, specifically for a specific number of genes, we must have the maternal gene that is activated. All of us. For other genes, certain genes, the paternal gene must be activated and the other must be inactivated. So for certain genes, mother's genes must be activated and father's would be inactivated. For another group of genes, the father's genes must be activated but not the mother's genes. Okay, so there is this interesting regulation of genes, and this phenomenon is known as genetic imprinting. So how can we inactivate the maternal genes or the paternal genes? Well, it's by methylating DNA at cytosines. Okay, so now this is epigenetics. Now look at these two twins. Okay, they are identical twins, right? Their DNA is exactly the same, yet they are different. I mean, you look at them and you notice that there are differences between them. Okay, why? They have the same DNA sequence. Yes, but they have different DNA modification and histone modification. So their epigenome really is different. Okay? Now, how does it become different? It becomes different because of their lifestyle. Meaning that it depends on what they eat, what they do. Do they play sports or not? Do they take care of themselves or not? Do they sleep early or not? Do they drink milk a lot or not? So this lifestyle habit would actually regulate the epigenome. So it's a higher level of regulation. Well, look at these, for example. These are twins again. These are twins. Yet you look at them and you can tell the difference between the two. And these differences are due to epigenome rather than changes in the sequence of DNA. Look at these mice. They have the same exact DNA, but their epigenome is different. And that's why they have different skin color. Okay. So the idea here is that epigenetics is really significant and it is heritable as well. In other words, what parents do would influence their children. So for example, if a mother smokes, that would affect children when she's pregnant with them. So if she takes care of herself during pregnancy, same thing with the father. Okay, so epigenetics can also be transmitted and it is heritable. So stress really can affect children, even though they would inherit the same gene, same sequence from parents, but the pattern of methylation or modification of histones and DNA would be different. So this is why Time magazine had this on the cover. That is, you really control your destiny. Okay, why? Because you may have a mutation, but you can suppress this mutation by leading a healthy lifestyle. So some women, for example, they have mutation in a gene known as BRCA1. We talked about BRCA1 before. BRCA1 is associated with breast cancer. So some women have a mutated BRCA1, yet they do not develop cancer, breast cancer. Why? Well, epigenome, epigenetics, leading a healthy lifestyle. So here's a scenario for how transcription factors function. And I'm going to use androgen receptor as an example. So, and we'll talk further about these steroid receptors in a second. So what androgen receptor does is the following. Okay, it binds to a ligand, to a hormone, to androgen, the male hormone. So the male hormone, the ligand, binds to the receptor. The receptor can then bind to DNA in association with a transcription factor, or let's say a co-regulator. يعني مساعد, a co-regulator. So they bind to DNA, and this co-regulator is actually an enzyme. What it does is that it is a kinase. That is, it's an enzyme that phosphorylates, that adds phosphate groups to histones. So what happens here is that you have phosphorylation. So that's phosphorylation of histones. You have phosphorylation of histones by this protein right here. Now, why specifically these proteins? Because it's led by the androgen receptor. Androgen receptor is activated, it binds to DNA, and it brings this to this particular region of DNA, so you have phosphorylation. Okay? Then, this would also attract this complex of proteins, two proteins, would attract another protein regardless of the name. And this here is actually a deacetylase. So what it does is that it removes the acetyl group right here. Okay, so right here, methylation. I'm sorry. So it's a demethylase. It removes the methyl group right here. Okay, so what it does is that you have the methyl groups then removed. Okay, and when methyl groups are removed, then you have activation of the gene. Okay, now you have transcription. The RNA polymerase, DNA becomes euchromatin. RNA polymerase can bind to DNA, and you have induction of transcription. Okay, so this is how transcription factors function. So right here, here's a little more detailed illustration of how it works. So right here, DNA is actually in the relaxed form. DNA is exposed, and these are the, okay, so I'm sorry. Okay, so right here you have a regulatory transcription factor bound to DNA at a certain sequence right here. Okay, that's a gene. Okay, transcription start site. You have promoter proximal element. Okay, and you have this transcriptional factor that binds to DNA. Now, when you have the formation of a pre-initiation complex, or sorry, when you have the formation of a pre-initiation complex, okay, bound with enhancers, proteins bound with the promoter proximal element, so you have this interaction right here. The DNA is actually now relaxed. All sequences are exposed. Then what happens is that the RNA polymerase would bind to the promoter, and you have induction of transcription. So that's basically how transcription factors function, by modifying histones and DNA. So here's an example of nuclear steroid receptor, like androgen receptor, for example. So now the idea is that these receptors, like androgen receptor, they have 3 domains. Remember what a domain is. So a domain is a 3-dimensional structure of a protein. Usually it has a function, and it folds independently, or it is independent of the rest of the protein. So if you remove it from the rest of the protein, it's still functional. Now, these steroid receptors have three domains. One is known as the ligand binding domain. This is the domain that binds to the steroid hormone, to the androgen. Another example is estrogen receptor, which binds to estrogen, the female hormone. And there is an activation domain that binds to co-activators or co-repressors, collectively known as co-regulators. And you have the DNA binding domain, okay, which bind to a specific sequence of DNA. So what happens here is that the hormone goes into the cell, right? It binds to the receptor. The receptor is now activated, so it binds to the receptor. That's the receptor right here. And you have the formation of a dimer of these receptors. The dimer goes into the nucleus, so that's the nucleus right here. It binds to a hormone response element. Remember how I said element indicates a sequence on DNA or RNA. So it binds to the hormone response element of certain genes, and then co-regulators would bind to the androgen receptor, and these would modify DNA or histones, relaxing DNA in case a gene needs to be expressed, or actually forming heterochromatin in case a gene needs to be repressed. Okay, so in fact, the thing is, you can have co-activators that can bind to estrogen receptor, as in here, for example, you can have activators that loosen DNA, inducing gene expression, or you can have co-repressors for certain genes, okay, that would actually repress expression of genes. Okay, so this is how genes in eukaryotic systems are regulated. Listen to this lecture again in order to understand it. Allah ma'akum.